Welcome everyone to the NPTEL course on Food Science and Technology, which is instructed by Professor H.M. Mishra sir, who is the professor in the Department of Agriculture and Food Engineering, IIT Kharagpur, and I am Shikha Sharma, research scholar in the Department of Chemical Engineering, IIT Guwahati, and I am the TA of this course. And this is the live interaction session for week 5. So in this, the assignment deadline for week 5 is 25th. So do submit on time. So the topics that we will be discussing in the today's class these topics are related to module 5. So it includes basically the food macronutrients and their structure and the function. So it basically will going to include structure of water and ice, free and bound water activity, and food stability, types, structure and function of the carbohydrates, then lipids and proteins, browning reactions, residity, and protein denaturation. So this live interaction session is basically we need to solve the question or the basically the theoretical questions which are related to this lecture 1 to lecture 5. Sorry, lecture 21 to lecture 25. So which are related to water, carbohydrates or the sugars, starch, cellulose and pectins, proteins and the polypeptides, and triglycerides and phospholipids. So which this all 5 lecture videos, I hope you have all gone through these videos. Now we need to solve some theoretical based question, and we will be discussing the reasons also uh, behind each question. And if you have any doubt, please feel free to ask during the class or at the end of the session. Also, you can post your questions on the Q&A. I hope I have given all the all the answers of your questions which were posted on Q&A or the discussion forum. So let's begin with today's first problem. So first problem is related to match the following. So it includes column 1, which is having the properties of water like high latent heat of vaporization, high specific heat, polar nature, and low density of ice. In column 2, we have the significance of those properties in the food system. So A is facilitates temperature and regulation in cooking. Second is essential for dissolving nutrients and salts. Third is prevents ice crystals from forming in frozen food structures. And D is makes evaporation and energy intensive process. So anyone who would like to answer which one is the correct answer out of all these? Meanwhile, I'll take the attendance. Okay. So let's see the answer of this question. So Hosea is saying A. Anyone else who would like to answer this question? Okay, let's discuss the solution part. So she's correct. Answer A is correct here. So we know that water basically it is a molecule that consists of 2 hydrogen atoms bonded to the sp3 orbital of an oxygen atom. So here we can see that here it is water co-molecule, which is oxygen, and there are two hydrogen atoms. So oxygen atom is attaching with two hydrogen atom. So the bond angle between the two hydrogen atom, because it forms a V-shaped structure. So the bond angle is around 104.5 degree, and the OH bond length is around 0.096 nanometer. So this V shape is because oxygen is basically because of the polar nature of the water molecule. So oxygen is basically having the partial basically negative charge, and water sorry H is having partial positive charge. So overall it will form the polar molecule. So water is an basically, since it is a electrically neutral molecule, there is an imbalance of charge because of the arrangement of atoms. So because of arrangement, it is basically since H is having 2 plus 1 plus because there are two H atoms and one is oxygen atom. So overall, if we see, that is 2 minus. So overall, basically, it is electrically neutral atom. So, but there is an imbalance of charges because of the arrangement of atoms in this, because we see that oxygen atom of the water molecule on one side is basically more electronegative, and the sides where the hydrogen atom are there, it is slightly more positive. So this basically going to induce the polar nature of the water molecule, which results in each hydrogen atom being attracted to the oxygen atom of another molecule, and the oxygen atom to the hydrogen atoms. So this basically type of the interaction or the attraction, it will going to lead to multiple hydrogen bonding on a three-dimensional basis. Each water molecule is able basically is able to form the hydrogen bond with maximum of 4 other molecules. So basically this is water molecule is basically, it basically has the tetrahedral structure, basically like this. It will form like this. In D figure, it is saying that it is having a tetrahedral structure. So here if we see, the polar nature basically is there for the water. So basically this polar nature of water is basically making it an excellent solvent, which allows it to dissolve in the various nutrients. It dissolve, like it basically act as a universal solvent. Also we say it a universal solvent. So it basically dissolves various nutrients and the salts, which is essential for biochemical reactions in food preparations and preservation. So basically 3 is related to B, which is it helps this polar nature of the water molecule. It helps in adding to, like it makes it a universal solvent in which different nutrients and the different salts, they can basically mix in the water, and that's why it's a universal solvent. Now let's see the ice. So irregular ice basically it is basically having dihexanal bipyramidal class of hexagonal system of the crystal symmetry. So it is like this. So this structure in the 3D lattice we can see the ice structure. So basically it can exist in 9 other crystalline polymorphic structures also, and also in an amorphous or vitreous state of uncertain but largely non-crystalline structure. And ice, when it is at 0 degrees Celsius, it is less dense than water due to, excuse me, someone is talking. Can you mute yourself? So we were talking about ice, that ice is basically having a hexagonal type of crystal system. So it is also having other crystalline polymorphic structures also, and it can also like sometime form the amorphous structure, uh, but mostly it will be in the crystalline domain, or it will form the crystalline structure. And ice at 0 degrees Celsius, if we see, it will be less dense than water due to more empty space in the ice. So we can see when it is here on the left hand side, we can see it is a water. So water molecules, they will have the loose hydrogen bonds between them, and basically it will form like this structure. But when we convert it into ice, like when we cool down or when we freeze down, so what will happen? There will be large gaps will form. So there will be open spaces between, like this. So lower between the molecule, inside the molecule, there will be basically more of the open spaces are there. So it is because of the formation of strong stable hydrogen bonds between water molecules at 0 degrees Celsius, which forms a basically rigid hexagonal crystalline structure, which we discussed that ice forms a basically hexagonal crystalline system. So it forms this structure. So because of this, it forms gap, and because there is a gap, so its density, the density of ice will going to be lesser than the density of water. That's why we see a floating of the ice on the water surface. So the low density of the ice is because of its structural property. So is significant in food systems basically because it helps prevent the formation of large ice crystals in frozen foods, which can basically damage the cell structure, and it negatively affects the texture. So if we see here, the mass the following. So the low density of the ice, which is the fourth one, is basically related to that we need to prevent uh, the ice crystal from forming in frozen food structures. Then water's high latent heat of vaporization means that a significant amount of the energy is required to convert water from liquid to vapor, making evaporation extensive, energy extensive process. So basically, the latent heat of vaporization of water means when we are supplying the heat, which is on x-axis, then we are supplying the heat, then how the state or the temperature of a particular material changes. So basically, when we supply the heat, so ice is becoming into water. So when the phase is changing, it is known as a latent heat of fusion. But when liquid is converted into gas, that is latent heat of vaporization. So temperature will be same here, but the state will change from the water to ice. So that is, sorry, water from water to gas. So that is latent heat of vaporization, because vapors are forming. The name itself is suggesting that latent heat of vaporization means vapors are forming. That means the water is converting into the vapor forms. But the temperature will be same here. And when we call latent heat of fusion, there what solid which is to turn into the water liquid. So this property is basically important in the cooling processes and in controlling the temperature during the cooking. And another is high specific heat. So specific heat is related to change in temperature, whereas latent heat is related to change in the phase. So here, if we see in the options, latent heat of vaporization is basically related to make evaporation and energy intensive process. So high specific heat is basically specific heat is basically if we have suppose water or anything, any metal like gold. So if we are increasing the temperature 1 degree Celsius, so how much heat we need to give to the system or to the product or to the material, so we can raise the temperature of that particular 1 kg of the substance by 1 degree Celsius. So its formula is Q equals to mCp delta T. So Q is basically the heat supply which we are supplying the heat. So Q is heat supplied is equals to mass, which is, suppose if we are saying, so how much is the heat we are supplying to convert 1 kg, if we are supposing it 1 kg. So 1 kg of the substance to delta T is 1 degree Celsius. If we are raising the temperature, or we are basically even we can decrease the temperature. So for altering, basically usually we take rise in temperature. So for rising 1 degree Celsius of the temperature, how much heat we need to require? So that is specific heat. So specific heat basically allows water to absorb and release large amount of heat without a significant change in the temperature. So which helps in temperature regulation in cooking, ensuring that there will be even distribution of the heat. So here in this question, high specific heat is basically related to facilitates temperature regulation in the cooking. So we are saying that option A is correct here. Is this question clear to everyone? Let's see how many people have attended today. Okay. Okay, we are moving to the next question. Which of the following best describes the effect of the solutes on the freezing point of water in food? So first is solutes increase the freezing point, solutes decrease the freezing point, solutes have no effect on freezing point, and D is solutes make water molecules uh, molecules form stronger hydrogen bonds. So if we are adding basically solute to the water, so what is going to happen with the freezing point? Anyone who would like to answer this question? B, Fozia is saying B, which is decreasing in the freezing point. Anyone who would like to answer this question? Okay, let's see the answer. So she's correct. that there is a decrease in the freezing point. So when we have the solutes, so it will going to basically influence both the amount, which is through the thermodynamic effect, and the propagation pattern also, which is through the kinetic effect of the ice in an aqueous system. So basically, we have the water, pure water, and one, if we are freezing it, converting it into ice. So this is water into ice we are converting. So when we are converting ice into ice, so if there is some kind of solute is present, it will going to affect both the amount and the propagation patterns of in the ice. Basically, so if we are like suppose we are adding solute in this water, so if we compare with the pure water, so its freezing point will going to lower down. So this phenomena is very well known as the freezing point depression, and the solutes which are interfering, basically it is going to interfere with the formation of the crystalline structure of ice. So we just now discussed that ice forms a crystalline structure. Suppose like this, here we discussed that ice is going to form this kind of structure, which is basically a hexagonal type of the system, where it will have this kind of three in the 3D lattice. If we see, it has hexagonal type of system, and it will be basically crystalline in structure. So it is having a proper uh, ordered structure. So when we are adding the solute, basically these solute molecules will going to interfere with the crystalline structure of the ice, and by disrupting the hydrogen bonding network. So in ice, we have the hydrogen bonding network through which the water molecules are connected. Solutes are going to disturb that. So this interference basically requires a lower temperature to achieve the same degree of molecular order needed for the water to freeze. So now, since solute is interfering with the structure of the ice, so we need more decrease in the temperature, or more lower temperature we need to freeze the water then. So because of that, we say that solutes decrease the freezing point. So the amount and the kind of solute, basically which how much concentration of the solute we are adding, and also how what is the nature of the solutes we are adding, that will basically affect the quantity, but also it will going to affect the basically size, structure, location, and orientation of ice crystals resulting from any particular cooling protocol. So how, which kind of like the solute particles we are adding, it will going to affect the overall property of the ice and the overall structure of the ice. So solutes basically, it will do not stabilize water molecules to increase the freezing point. Instead, they are going to destabilize the hydrogen bonding network. So that's why it's disrupting the freezing point. So the solute do indeed basically affect the freezing point. It affects the freezing point. Also, it unstabilize the water molecule structure. Then solutes also do not strengthen the hydrogen bonding. They actually disrupt these hydrogen bondings, which will lead to lower of the temperature to freeze the solution. So if we discuss here, so it is saying that the solute increases the freezing point, which is wrong. And then solutes have no effect on freezing point. So it is, and the solute makes molecules form stronger bonding, which is also wrong because it disrupts this hydrogen bonding network. So if we see from the phase diagram, so here if we see, when we are freezing, this is the pure solvent. So pure solvent, if we are freezing, so it's when we are decreasing the temperature from here, like from when we are decreasing the temperature from here to here, so it is like that. But when we have added some kind of solvent, so its freezing point basically it shifts. So this change in the freezing point, now this is the freezing point. So we can see that this is the change in the freezing point. So this is delta TF. So this depression in freezing point, basically it can be calculated by this formula, which is equals to I Ventov factor into K, which is its uh, constant, and into molality. So this is the freezing point depression formula. So this we have already, this type of formula we see in 11th, 12th chemistry also. Okay. So is this question clear? Can we move to this next question, which is about which of the following conditions will inhibit microbial growth in foods? So water activity above zero point 0.9, water activity below zero point 0.6, low ambient humidity, or water activity between 0.7 to 0.8. Anyone who would like to share his opinion on this question? Shivaswamy and Fosia are saying B, which is water activity below zero point 0.6. Okay, let's see. So they are correct that microbial growth in food basically affected by the water activity, and water activity is a measure of availability of the water which is for the microbial growth or any enzymatic reaction or any biochemical processes. So the water activity is basically the ratio of the, this is P basically by P naught. Water activity is the basically the ratio of the vapor pressure of the food when in a completely undisturbed balance with the surrounding air media and the vapor pressure of the distilled water under identical conditions. So it is water activity is basically P by P naught. So we can convert it into equilibrium relative humidity by multiplying water activity into 100. So if we multiply water activity into 100, which is P by P naught into 100, so we can get equilibrium relative humidity. So basically this water in basically in the food, it exerts a vapor pressure, and the size of the vapor pressure basically depends on the amount of the water present, the temperature and the concentration of the dissolved solutes like the salts and the sugars which are there in the water. So if we see the range of water activity, so if water activity is very high, which is 0.9121, the bacteria can easily grow, and water activity 0.88 to 0.91, yeast can grow, and water activity 0.7 to 0.88, moles can grow here. So water activity between 0.7 to 0.8 can lead to growth of the molds, and if it's greater than 0.9, bacteria can grow. Also, like if it's greater than 0.7, yeast can also grow. So we need to decrease the water activity below 0.6. So most bacteria, it requires water activity level above 0.9 to grow, while the mold and the yeast, they can survive at the lower water activity like 0.7 to 0.8. However, when the water activity falls below 0.6, the environment becomes too dry for the most of the microorganisms to sustain growth, effectively inhibiting the microbial activity and the spoilage. So basically, if water activity is less than 0.6, microorganisms will going to inhibit. So next is ambient humidity. Basically, it does not directly impact water activity within itself, making these conditions less effective at inhibiting microbial growth and compared to the water activity below 0.6. So if we see here in this diagram, so if water activity is less, like less than 0.6, there is no growth of mold, yeast or bacteria, but we can see enzymatic activity still be can happen. Also, the non-enzymatic brownings can also happen. Lipid oxidation can happen, and hydrolytic reactions can also happen. So it depends, like how much is the water activity we have. So water activity which is around 0.2, 0.3, most of the things will going to be halted. So we need to basically maintain water activity such that the growth of mold, yeast and bacteria will inhibit. So like this, we are keeping 0.6. So their growth is inhibited and little bit enzymatic activity we can observe. Also, we can observe here other reactions like browning. So but what happens if we have the too low water activity? So the texture of the product will going to hamper, like there is a loss of crispiness or there can be collapse of the structure of the food product. So basically we need to make the water activity level in the optimal range so that the inhibition of the growth of the microorganisms can be seen, but simultaneously we can maintain a good amount, good texture or good mouthfeel or good sensory attribute, overall sensory attributes at the same time. So we need to maintain such water activity. So here answer is B, which is water activity below zero point 0.6. Let's see this next question. The sorption isotherm represents the relationship between relative humidity and which one? Water activity, temperature, moisture content, vapor, pressure. Anyone who would like to answer this question? Okay, so answer B and C. Shivaswamy saying B, Hosea saying C, either temperature or moisture content. Anyone else who would like to answer? Okay, here C answer is correct, which is moisture content. So basically, sorption isotherm graphically represents the relationship between the relative humidity of the surrounding environment and the moisture content of a food product at the constant temperature. So basically, at constant temperature, we'll have relative humidity on the x-axis, and on the y-axis, we'll have moisture content. And the plot will going to give the sorption isotherm. This plot is basically known as sorption isotherm. So it tells the behavior of the absorption and desorption of a material. So which means that basically it tells about how much of the moisture basically a food is absorbing or desorbing or desorption, or it is going for the desorption at different humidity levels. So basically it has two parts, which is adsorption isotherm and desorption isotherm. So in adsorption isotherm, which is this one, so how water is adding, and here desorption, how water is losing. So basically it is going for adsorption isotherm is for moistening the food. So moistening of the completely dried product by placing into various atmospheres of increasing relative humidity. So we're going to increase the relative humidity and we'll see how much is the moisture or the how much is the water the completely dried food is taking, and we can measure the weight gain which is because of the water uptake. Whereas in desorption isotherm, we have the wet food or the moist food, and we're going to dry it under the same relative humidity condition, and we will measure what is the weight loss or the water loss. So we can plot. So if we see here, so when we are increasing the water content in the food, and when we are removing or we are drying the food product, so it will not going to follow the same path. It will going to have some kind of this gap which is known as hysteresis. So the difference between these curves of adsorption and desorption isotherms, it is called hysteresis. So while water activity basically here, so this is the moisture content is the correct answer. So if we talk about the water activity, so water activity basically is related to the moisture content, and it influences the microbial growth and the chemical reactions. The sorption isotherm basically, specifically it is for the relation between the moisture content and the relative humidity. This water content, water activity is going to relate, it is related to the moisture content term, but the if we talk about the sorption isotherm, it is basically having this moisture content relation with the relative humidity, not water activity. And we know that temperature is going to affect the sorption isotherm and its shape. But is not the variable plot on the graph. So at constant temperature, we measure this graph between the water or the moisture content with the relative humidity. Uh, so if we change the temperature, obviously the pattern of this adsorption and desorption will going to affect or change. But on the same time, if we see, we measure this graph, or we plot this graph at the constant temperature, and we take the readings of the, like, we vary the relative humidity and we measure how much is the water is absorbing or dissolving. So uh, relate water activity is not correct, temperature is also not correct. And let's talk about vapor pressure. So vapor pressure is basically related to water activity, because we take vapor pressure P by P naught in water activity, but it does not directly describe the sorption behavior. So if we see here this plot, so it is the adsorption and this is the desorption. In between is the gap, or the difference is called as the hysteresis. So in the starting, if we see, this water is basically the strongly bounded water on in the monolayer. Like if we have this food product, so the strongly or the closely bounded water molecules will be there initially. Then next is the layer of the water molecule which is less strongly bound water, and like this, in this particular region, third region, is the water which is free water. So it will, the free water will be like, it will be easily basically going to be, this free water is going to be easily absorb also and easily dissolve also. Whereas if we see the capillary adsorb water, or the less, or the second one, moderately bound water, it will not, like, it is not that free also, but it is not strongly bounded also. But if we see the strongly bounded monolayer, it will not be very, like, removal of this is very difficult also. So this is very, like, because it is in the close proximity with the food product. So there are different 5 type of the adsorption isotherms available. So we classify adsorption isotherms, which is this graph basically. We classify into 5 different types. So one is when the ratio, like this is P is the vapor pressure and X by M, like this is the, we are taking the Langmuir's equation. So in this, if the, suppose if the what, like the, there is a mono layer is there in the Langmuir adsorption isotherm. So basically it will going to follow this kind of the pattern. So what will going to happen if we increase the uh, uh, vapor pressure? So what will going to happen? The monolayer will going to form, and after monolayer will form, it will, we can see the saturation one. So next is a type 2 adsorption isotherm. So in this, what is going to happen is, in this there is this flat initially the water will going to be absorb, and this until this is the same like monolayer formation, but then there is this increase, sudden increase, which is, we can see there is a sharp knee kind of formation, which represents the formation of the multilayer. So water first it will go absorb like monolayer formation, and it will occupy all the surface of the food. So till the absorption increase, and once the surface is covered layer of the water, it will be saturated. So in type 2 types basically the multilayer also. So once the monolayer is formed, then the sharp knee is going to depict the multilayer formation. So in this third one is also depicting that after the monolayer adsorption is done, there is a sharp knee formation which represents the multilayer formation. And if we see this type 4 and 5 graph, so it is basically depicting, this one is basically depicting that at lower pressure, basically it is increasing and monolayer is forming, the adsorption is increasing and the monolayer is forming, and after the saturation level reach at this particular temperature, at particular pressure, vapor pressure, so then gas will going to be just like this. If there is this particular food product, so there will be different pores inside it. So the water molecule will go and going to be condensed in these capillaries, tiny capillaries, and because of that there is saturation is there. So that's what we can see here. Also this is also representing that this is the basically capillary condensation. So overall what happens is, when there is any food product, so I'm summarizing that if there is any food product about adsorption, so what will going to happen, it will have some kind of pores inside it, right? Like this, like this. So what will going to happen, first water will come and adsorb on its surface like a monolayer. So once the all the surface will fill, then the next layer, which is second layer, will form of water molecule, which is known as multilayer formation. Okay, and after this multilayer will form, then what will happen? This, in this space also water molecule will go and it is going to condense here, which is known as capillary condensation. So it is going to condense here. So like this adsorption phenomena takes place. So in this question, the answer is moisture content, that the relationship between the relative humidity with the moisture content is sorption isotherm. Is this question clear to everyone? Can we move to this next question? Match the terms related to water in food, which is 4 different terms: bound water, free water, glassy state, and water mobility. In second, their definitions are given. like water molecules trapped in the food matrix and unavailable for the reactions. B is state whether molecular movement is minimal, enhancing the stability. Third is water that act as a solvent and is easily extracted. D is movement of the water molecules affecting the reaction rates and stability. So Shivaswamy and Fozia are saying C. Okay, let's see. So they both are correct. C is the correct answer. So bound water basically is going to refer to the water molecules that are tightly associated with the food compounds, making them chemically inactive and unavailable for the reactions like microbial growth and chemical transformation. So this bound water is will be going to be in very close proximity with the food components or the food particles. So it exists basically in the vicinity of the solutes and other non-aqueous constituents, and that are a result of its location, exhibits apparent properties that are significantly altered from those in the bulk water in the same system. So this bound water will going to be, because it's in the close proximity with the food, so it will going to have some different property, a little bit different or deviated property as compared to the free water or the water which is present in the bulk. So it is stable water basically of the sample at a specific temperature and low humidity. So when at a specific temperature and the low humidity, it is basically the stable water content. So bound water is related to water molecule which is trapped in the food matrix, and basically it is unavailable for the reactions. So it will basically, since it is very much in the close proximity with the food or the solute particles, so it will not going to be available for any kind of the reaction. So it is a chemically inactive water. So this bound water, it is basically unfrozen state, so it will not going to be freezed even at minus 40 degrees Celsius. So at minus 40 degrees Celsius also this will not going to be freezed. Otherwise, if we see the free water, that free water will going to be freeze at 0 degrees Celsius, right? So that because it is in the close proximity with the food components, so its property is very changed. So the bound water is basically the water that moves with macromolecules during the sedimentation, viscosity, or diffusion experiments. It is very strongly bound with the, like this, if we see here in this diagram, bound water is strictly like the close proximity with the solute, and the free water is available for the microorganisms because it is away from the solute. And even if we see here in the this diagram, the free water is the one which is available here, and the bound water is with the food product. So the free water is a water that can act as a solvent and is easily extracted and is readily available for the chemical and the microbial activities affecting the food texture and spoilage. And we can get this water by pressing, squeezing, or cutting the food product. It is a water that is not chemically bound to other materials and can move or be extracted easily. So it has a high vapor pressure and can freeze at moderate temperatures. So if we talk about the free water, it is a water that acts as a solvent and is easily extracted. Now, let's talk about the water mobility. So, food systems basically they are complex mixture of water, biopolymers, ingredients, colloids. So, all are there in the food system. So, where the molecular mobility basically going to influence the stability, physical state, microstructure, and food characteristics. So, food water content, basically its location and its interaction with other components is very important to understand the microbial growth, degradation, and the different uh, enzymatic and the non-enzymatic degradation reactions and the sensorial attributes, so like the texture or the juiciness or like that sensory attributes. So water mobility is basically it is related to the movement of the water molecule in the food product, which will going to affect the reaction rates and overall food stability. So like oxidation reaction is there, Maillard browning is there, phase transition is there. They all depend on the molecular mobility. Temperature and moisture levels, basically it affects the molecular movement influencing the stability. So if we see here, the water, like this is a cell wall and this is the basically little bit space is there. So water is basically move across this transport in this the tissues or the apple tissues here shown in the figure. So here if we see water mobility, basically it is related to the movement of the water molecule affecting the reaction rates and the stability. Now the glassy state. So glassy state is basically it is a condition where the molecular movement in the food matrix is minimal. So in the like if we see here in this diagram, so the glassy state is the one where there is low mobility, and because of the low mobility, higher stability is there. So the food product often looks like the solid like. So it enhances the stability of the food by reducing the rate of the chemical and the physical reactions. So the mobility, it affects basically the texture, crystallization, and phase separation, which are critical for the maintaining the food quality. So this is the state diagram in the food product. So here on the y-axis, there are the water content, and on the y-axis is the temperature. So if we see here, under different temperature and moisture conditions. So there is this glassy state. So in this glassy state, what happens is there is low mobility of the moisture, and because of that, there is a higher stability of the food product. Then second is the rubbery state. So rubbery state, in the rubbery state, little bit mobility increases, so it becomes less stable and it will be little solid like or flexible. And third is the crystalline state where there is organized structure we can see, and the more stable systems we can see. And another is the glass transition line. So there are different critical lines we can see here in this state diagram. So glass transition line, it separates basically the glassy to rubbery states, and melting line, it indicates the transition between the solid and liquid phases. So here, third one is the glass state, which is related to state where molecular movement is minimal, so enhancement of the stability. So answer C is correct here. Okay, let's discuss this question. Which of the following statement about the glass transition temperature is our correct? Statement 1 is saying that the TG, which is glass transition temperature, transition from a rubbery to a glass, and food, second statement says foods are stable below their Tg due to the increased mobility. Anyone who would like to answer this question? Okay, so we have 4 answers right now. So Srivaswamy A, only one. Aboli is A, Fozia A, Aman is B. Okay. So 3 of us are saying A, Aman is saying B, Anjali is saying C. Okay, let's see. This is the correct answer. So D is the correct answer, that both of the statements are not correct here. Why? Let's see. Because glass transition temperature, it is basically, it marks the transition from a glassy or the rigid state to a rubbery or more flexible state, which is here written reverse. So this statement is wrong because it is depicting the glass change of the glassy to the rubbery state. If we see the polymers also, if we do the DSC analysis, so here is the heat flux is there, and here is the temperature is there. So we can see here is a glass transition state. We can see in the polymers also when we heat it or we undergo the DSC or the differential scanning calorimeter analysis. So what happens there also, there is a glassy to rubbery state we can see transition. So below Tg, food materials basically they are in the solid like or the glassy state where the molecular mobility is significantly reduced, leading to enhanced stability and a decrease in reaction rates such as lipid oxidation, enzymatic activity, and microbial growth. So at low Tg, so food is become more stable because there is less mobility. So which this statement second one is also incorrect here. So as we already discussed in the previous question also, that we have this glassy state to rubbery state, and this transition is from the glass transition line. So we can see here also. And here, since when there is a low Tg, that means glassy, so it glassy, there is low mobility and stability. I hope this question is clear to everyone. Let's discuss about sucrose. The systematic name for sucrose is, out of these 4, please tell me the systematic name of sucrose. Shivaswamy C, Fozia A. Okay. So A is correct here. So the systematic name for sucrose is O alpha D glucopyranosyl 1,2 beta D fructopyranoside. So basically it describes basically the glycosidic linkage between the glucose and the fructose. So if we see the here the structure, so it is a glucose and fructose, and in between there is a glycosidic linkage in the sucrose. So the sucrose basically is a disaccharides which is composed of glucose and fructose and are linked with alpha 1,2 glycosidic bond. And the term O alpha D glucopyranosyl, it indicates that a glucose unit is in the pyranose, which is 6-membered ring form and attached to the oxygen atom of the anomeric carbon in the alpha configuration. So if we see here, it is alpha D glucopyranose because there is the 6-membered ring and oxygen is attached here. Next is fructose. So the beta D-fructopyranoside, basically this portion is going to specify that there is a fructose unit in the is there in the furanose, which is 5-membered ring form and are attached to the again oxygen atom of its anomeric carbon in the beta configuration. So here there is a 5-membered ring and oxygen is attached here. So the number 1, 2 here. So here we understood this part and then this part also we understood. Now 1, 2 is depicting basically the number 1, 2 denotes the carbon involved in the glycosidic bond with carbon 1 of the glucose linkage to the carbon 2 of the fructose. So here carbon 1 which is here is attaching to the carbon 2 of this fructose. So basically this sucrose, it is a colorless crystalline substance which is sweet in taste and it is soluble in water, and the solution is basically dextrorotatory, which is D plus 66.5. When heated slowly, sucrose can melt and it will solidify and it will convert into pale yellow gloss mass, which is called barley sugar, and you at higher temperature, which is 473 Kelvin, it is going to dehydrate completely and basically a brown amorphous mass we can obtain, which we known as caramel, or we can caramelize the sugars with a strong heating, and it at the end it will form a charge to nearly pure carbon, producing the characteristic smell of the burnt sugar. So is this question clear to everyone why we call sucrose as this? Okay, let's move to the next question. Which of the following is not a reducing sugar? Glucose, fructose, sucrose, and maltose. Shivaswamy D, Fozia C, and Aman A. D, C, A. Okay, let's see. Correct answer. So basically C is the correct answer. Sucrose is a non-reducing sugar because it does not have a free aldehyde or ketone group which can going to participate in the redox reaction. So if we see here in this diagram, so basically we can see that sucrose is not having any aldehyde or ketone group attached, which is free basically. Free aldehyde or ketone group is not there. So if there is no free aldehyde or ketone group, so it cannot participate in the redox reactions. So in reducing sugars like glucose, fructose, and maltose, there will be going to have one anomeric carbon which is free, and it can act as a reducing agent. Like if we see in this diagram, so this fructose, this is the fructose. So in fructose, glucose, and maltose, we have free anomeric carbon groups which will going to participate in the reactions of reduction or of the redox reaction. So we can have basically in sucrose, glycosidic bond between the glucose and the fructose, it involves both of their anomeric carbons. So because of that, there is locking of the reducing group. So because of that, the prevention from the oxidizing reactions or the as an oxidizing agent. So it cannot basically react, this sucrose cannot react with oxidizing agents, and it cannot act as a reducing agent. So sucrose basically cannot reduce this solution. That's why it is known as non-reducing sugars. Basically, this is the anomeric carbon here. The anomeric carbon here. It is not here in this glucose and fructose, it is free, but here it is forming a bond. So we cannot go and we cannot form, like we cannot go with the sucrose as an oxidizing, or sorry, as an reducing agent. So basically, sucrose, it is basically this, or this is the non-reducing sugar, right? But there are different reducing sugars also. So like the glucose, fructose, or the maltose. So what test we can from which test we can confirm that our sample is having glucose or the fructose or the maltose or any kind of reducing sugar? So there is one test known as Benedict test. So what happens here? So in Benedict test, in the test tube, we'll take the food sample in which any kind of sugar is there. In that Benedict reagent, we will going to add, and then we'll heat this uh, test tube for at the boiling point for 5 minutes. So if we see the change in the color, so if there is presence of the reducing sugar, so if the high amount of the reducing sugar is there, we can see the red color. And if there is no reducing sugar, we can see the blue color. Or if we can see, so this color will be with higher concentration, it will be more red, from blue to green to yellow to brown to red. So it will be more red if more amount of the reducing sugar is there. Because what happens, the monosaccharides which are having the which are the reducing sugars, so they will have the carbonyl group. This group will be there, right? So what will happen, this will going to oxidize and it will form carboxylic acid. So in Benedict test, D-glucose is going to oxidize and it will form D-gluconic acid, and thus the glucose is considered to the as a reducing sugar. Also, like in Benedict test, what happened? Reducing sugars, they will heat in the presence of an alkali, and it will get converted to powerful reducing species like inidols. And when Benedict reagent will going to react with the sugar, it will and heat, it will change to the color of orange red or the brick red. So here we can see there are other tests like Toland reagent test or the Fehling solution also to determine about the reducing sugar. So if glucose, basically it get oxidized to gluconic acid with the ammonical silver nitrate in the Toland's reagent, and in the Fehling solution it will be uh, oxidized to gluconic acid with alkaline copper sulfate. So basically, Toland reagent is basically is reduced to metallic silver or silver Miller, whereas the Fehling solution to cuprous oxide, which is a red precipitate. So basically, what it is saying is, if we add glucose to the Toland's reagent, so what will going to happen? This is a glucose, and it is we are adding to the Toland's reagent. So what will going to happen? This reaction will take place and the oxidation of the glucose will be happening, and glucose will going to convert into gluconic acid, and this Tollens reagent will going to be converted into silver. So if there is negative tolerance test, means there is no aldehyde or the ketone or there is no reducing sugar, so its color will be like this. But if there is aldehydes or the ketones or the reducing sugar will be there, the color will going to be dark gray or the silver mirror kind of color we can obtain in the solution. Whereas in the Felling's solution, what will happen? Again, the glucose is going to convert into gluconic acid by the oxidation reaction, but the Falling solution in which there is copper, so that copper will going to be converted into cuprous oxide and it will form the red precipitate. So if we can have this red precipitate, so what if initially the color will be like blue, and when when the Felling's reagent is acting and the cuprous oxide is forming in the reducing sugar, the color will going to be reddish or the brownish color in case of the glucose or the fructose or any kind of reducing sugar. Okay, I hope so far is clear to everyone. Let's move to this question. Next question. Which of the following polysaccharides is the storage form of glucose in plants? So this is cellulose, starch, hemicellulose, or glycogen. Anyone who would like to answer this question? Okay, A, B, A, B. Okay, we have two doubts as starch and cellulose. Okay, so B is the correct answer. Starch is the correct answer here. So starch is basically the primarily storage polysaccharides in plants, and it contains two units, amylose and amylopectin. So amylose is basically the linear polymer and amylopectin is highly branched polymer. So it basically, this starch serve as a reserve for glucose in the plants and used for the energy during the periods of low photosynthetic activity such as the night time or unfavorable environmental conditions. So starch granules basically are stored in plant tissues like the seeds, tubers, and the roots, and it is going to basically act as a storage polysaccharides. Whereas the cellulose is a structural polysaccharides and it is found in the plant cell wall. So it is in the cell wall of the plant. So it is going to provide the structural framework and provides the rigidity and the support. Whereas if we talk about the hemicellulose, it is also contribute to the structural framework of the plant cell and is not used for the energy storage. And the glycogen, it is basically having more of the amylopectin and is basically act as a basically glycogen is similar to the amylopectin and it is also act as a storage uh, storage polysaccharide storage form of the glucose, but in the animals and the fungi but not in the plants. So basically we discussed the starch is composed of two polymers, linear is the amylose and highly branched is the amylopectin. So basically amylose, it is basically a linear chain of 1,4 linked alpha D glucopyranosyl units. And here we can see this is the amylose molecule. So some amylo molecules, they have the branches which are connected to the alpha D16 linkage. So the branch points are there, but very less, around 0.3 to 0.5%. Usually these amylose are linear in nature. It is not in branched in nature. Amylose adopt basically right-handed helical shape with a hydrophobic hydrophobic or lipophilic interior and hydroxyl group on the exterior side. So basically, the hydroxyl group will be the exterior side and the hydrophobic group will be in the interior side. Like that, it will form. So each helical turn, it will contain approximately 6 alpha D pyranesyl unit. So starch generally contains around 25% of the amylose and higher amylo corn starches contain around 50 to 75% of the amylose. Second is the unit is the amylopectin. So amylopectin is basically highly branched molecule. So it will have the branches around branch with branch points around 4 to 5% of the total linkages, and it is like this. So it will have this branch here attached with this linear chain. So the molecule consists of the main chain with reducing end groups and multiple branch chains forming the clustered double helix. So the amylose, it is basically having the high molecular weight compounds ranging from 10 to the power 7 to 5 to the power 5 into 10 to the power 8, making it one of the largest molecule in the nature. It consists of around 75% of the most common starches, and starches they mostly made up of the amylopectin, and with if the amylopectin amount is more, or if amylopectin amount is 100%, it will be waxy starches. Also, the potato amylopectin is unique for having phosphate ester group attached at O6 and O3 positions, occurring about once in every 215 to 560 alpha D glucopyranosyl units. So basically this starch is made up of two units, amylose and amylopectin. Amylose is the linear one and amylopectin is a branched one, and most of the starches contain around 25% of the amylose and 75% of the amylopectin. Then the cell wall compounds, which are basically the pectin, cellulose or the hemicellulose. So basically pectin is composed of the galactouronic acid. Here we can see. So it has ramenose, galactose and arabinose. Like this is the structure of pectin. Mostly its monomer is galactouronic acid, and cellulose is basically homopolysaccharides of glucose. So like this is cellulose, and hemicellulose is basically the main branch of xylox, in which arabinose and glucuronous acids molecules are basically attached. Glucuronic and xylose and arabinose. So three molecules are there basically in hemicellulose structure. If we talk about cellulose, so cellulose is consist of basically around 25% of the cell wall in higher plants and accounts for nearly half of the bird's carbon supply. So it is basically found in the woody tissues as partially crystalline microfibrils. In vegetable pulp, it has little fibrous characteristics. So it also embedded in an amorphous gel of hemicellulose and pectic substances. It influences the texture and the texture of the fruits and the vegetables during ripening, storage, and processing. It composed of long chains of glucose residues with beta-1,4 linkages, giving it a very high molecular weight. It is basically insoluble in water and not digestible by humans. So cellulose basically act as a dietary fiber or the roughage for human beings. It can be broken down by glucose by the microbial enzymes. So it can be digested by the uh, some of the animals, but cannot be by the humans. Hemicellulose, they are basically the having the group, a diverse group of the polysaccharides found in land plants, excluding cellulose, starch, and pectic substances. So it has basically xylose like that. So it's composed of the like pentose, like xylose, then hexuronic acids, and sometimes ramenose. And it is also insoluble in water, but it is soluble in alkali. Its function is to cement the cellulose microfilaments together, contributing to plant cell wall structure. It forms part of undigestible carbohydrates and it also serves as a dietary fibers. Last is the glycogen. So glycogen is basically the storage polysaccharides which are present in the animal. So it is known as animal starch. Predominantly, it is found in the liver and skeletal muscles. Rapidly, it will hydrolyze to D-glucose after slaughtering. So this is the glycogen. It resembles amylopectin, but it is more branched even than the amylopectin. So it is even more branched than amylopectin. Like this, there will be different linear chains and the branched one. So in this answer is starch, which is a storage form of glucose in plants, and in animals it's glycogen. Cellulose and hemicellulose are basically the cell wall structure. It presents in the cell wall of the plant. Okay, this question is very related and very simple now. Which of the following statement about amylose and amylopectin are correct? So statement 1 is saying amylose is a linear polymer while amylopectin is a highly branched one. And statement 2 is saying both amylose and amylopectin, it contributes to starch retrogradation. Anyone who would like to answer this question? So we have three answers, A, B, and C. So Shivaswamy is saying A, Fozia C, Aman B, and Anjali C. So we have mostly this answers here. Okay, let's see now. So C is the correct answer, that both statement 1 and statement 2 are correct, because as we just now discussed that amylose basically is a straight chain polymer while amylopectin is a branched chain polymer. So this statement 1 is correct. Now basically amylose is a linear structure, so it allows to pack closely together, which contributes to the starch retrogradation. So retrogradation of the starch is mostly contributed by amylose. But retrogradation is when cooked starch going to thicken and firms up as it cools, like when in the bread staling case. So both amylose and amylopectin, they are going to involve in the process of amylose, in the process of retrogradation of starch, though amylose basically has a stronger effect due to its simpler structure or straight chain it is having, whereas the amylopectin is having the branched structure, so it makes it less likely to form tight bonds. So basically since both contribute, amylose and amylopectin, both will going to contribute in the starch retrogradation, but the amylose is highly contributing towards it. So here both statements are correct. Is this question clear to everyone? Can we discuss this question? That factors influencing the starch retrogradation is listed in column 1, like amylose content, water content, temperature, and storage time. In column 2, it is written their effects like promotes crystallization at lower temperature, high content accelerates the retrogradation, adequate amount required for gelatinization, and T is the increase the firmness over time. Shivaswamy is saying A. Aman is saying D. So Shivaswamy is correct here in this case. So what is happening is starch retrogradation. Basically, it is a process where gelatinized starch molecules, particularly the amylose and the amylopectin, they realign and they form the crystalline structures over time, which affects the foods like bread and the rice. And amylose having the faster rate of the retrogradation because of its packed structure, which we just now discussed. So there are the different, if we see the structure, so amylose and amylopectin. So when it get the heat and the water, it gelatinizes, and after some time, the retrogradation process start. And in that, there is again the restructuring of the starch molecules will be there. So if we see here, with respect to viscosity, with respect to temperature in the x-axis. So with respect to increase in the temperature, we are seeing the viscosity. So we can see starting, there is like the gelatinization starting is there. And after that, there is a maximum gelatinization and maximum viscosity it can be achieved. After achieving the maximum viscosity, if we further increase the temperature, there's a complete disruption or completely dispersion will be observed. And after that, at this point, there is a starch retrogradation or recrystallization at higher temperatures. So this we can see here. So the factors which influences the retrogradation process are amylose and amylopectin content. So if there is a higher amylose content, it promotes the faster retrogradation due to its linear structure, while the amylopectin is having the branched structure, the process will be of the retrogradation will be slower, which we discussed already. Then water content will adequate water content is crucial for starch gelatinization and retrogradation, both. So too little water limits retrogradation, while excessive water dilutes starch chain, reducing the crystallinity. Then temperature, it lowers, so the lower temperature favors the formation of crystalline nucleus, while higher temperature promotes the growth of the crystals. And storage time, if we increase, the retrogradation will increase over the time, and short-term retrogradation is driven by the long-term retrogradation is dominantly uh, done by the amylopectin. Food additives like ingredients like lipids, proteins, and the sugars can either promote or inhibit retrogradation depending on their nature or their interaction with the starch. And the processing conditions like rate of cooling, retrogradation pattern, and the other environmental condition affect how the starch chain rearrange into a more ordered structure. So amylose content is related to the higher content accelerates the retrogradation. Then water content is basically if we have the adequate amount required for gelatinization. Temperature is related to the promotion of the crystallization at lower temperature, and storage time, it increases basically the firmness over time. So the starch will going to be, when the starch retrogradation will take place with the time, as we increase the time, starch retrogradation will also be more. So the more firm texture we can see. So I hope this question is now clear to everyone. So if we see the staling of the bread, so what happens? What starch retrogradation do? Basically, starch retrogradation can negatively impact the quality of starchy foods by altering their texture, flavor, and nutritional value. So we can see that bread staling is one of the example where this crumb firmness is going to increase and the fresh perception is going to be decreased because of that. And baked goods, starch basically transition from the amorphous to crystalline region. It states post baking. And shelf life, the surfactants like GMP, which is glyceryl monosaccharides, and SSL, which is sodium stearoyl lactylate, are basically added in the bread to increase the shelf life of the bread. And the retrogradation, basically it lowers the shelf life of the starchy food. So if we see here this bread, so bread dough basically it will become more crystalline will be there. So when after that it will gelatinize, and because of gelatinization starch will go into amorphous structure. But when there is a staling of the bread, again recrystallization will going to happen in the structure. I hope this question is clear. This until here all the questions are clear. So let's discuss this question. High methoxypectin requires which condition of temperature and sugar concentration, or pH, or about the ionic condition, or enzymatic activity, which will going to form gel. So because we know that the pectin is going to form gel like structure, and we can see different food products over the shelf in the market, these jellies like these products, so they are basically made from the pectin, and this is basically the result of the pectin structure. And in this we can see here in the red region there is a hydrogen bonding. So this linear chains of pectin, they will going to form the gel like structure because of these hydrogen bondings. So we need to tell which condition is favorable for the high methoxypectin to form gel. A, C, and D. We have three answers. Okay, let's see. So A is correct answer here. When we have high methoxypectin, so we need low pH, which is around 2.8 to 3.5, and a high sugar concentration around greater than 55% to form a gel. So when we have the low pH, it will going to ensure that the carboxylic group of the galactouronic acid unit, so this is the monomer of the pectin, so its carboxyl group are going to be protonated. So reducing the electrostatic repulsion between the pectin chains and allowing them to come closer. That's why they can form the gel-like structure. If there will be less repulsion will be there, then only gel can going to be formed. So the higher sugar concentration, it will help to dehydrate the pectin molecules and creating a favorable environment for hydrogen bonding and hydrophobic interactions, which will be essential for gel formation. So other options like B, C, and D, they are incorrect because high methoxypectin do not gel at high pH and low sugar concentrations, and they do not rely on temperature, enzymatic activity, and low ionic strength for gelling. So we need high sugar concentration and low pH to form a pectin gel. Let's discuss this question. We have in column 1 type of proteins, and in column 2 we have the example. So hydrogen bond, ionic interaction, hydrophobic interaction, and disulfide bonds. In column 2 we have the different examples of these bonds like cysteine linkage. B is stabilization of alpha helix and beta sheet structure. C is attraction between positively and negatively charged side chains, and D is clustering of non-polar side chains away from water. Anyone who would like to answer this question? B, everyone is on B. So you guys are correct. So protein interactions basically is important for maintaining the structure and function of the protein. So we know that the protein maintain different kind of the conformation, like with the help of the covalent interaction like the disulfide bonding, and non-covalent interaction which majorly includes hydrophobic interactions, and then electrostatic interaction, hydrogen bonding, and van der Waals interactions. So the hydrogen bonding is basically very important in stabilizing the secondary structures like alpha helix and beta sheets by forming between the backbone of the carbonyl, oxygen, and amide hydrogen atom. And ionic interactions, they occur between the side chains with opposite charge such as the lysine and the aspartate, and are important for the stability of tertiary and quaternary structures. So for secondary structures like helix structure and beta sheets structure, we need hydrogen bonding majorly, and for maintaining the tertiary and quaternary structures, we need to have this ionic bonding we should have. Then hydrophobic bondings or interaction, it involves the clustering of non-polar side chain like those in the valine and leucine and away from the water. So driving the folding of protein into a compact structure. So this will going to help to form the foldings, these hydrophobic interactions, and disulfide bonds are the covalent linkages formed between two cysteine residues, creating a strong bond that stabilizes the protein tertiary and quaternary structure. So that is in between two cysteine molecules. So hydrogen bonding is related to stabilization of alpha helix and beta sheet structures. Ionic interaction is for the attraction between positively and negatively charged ions or the side chains. Disulfide bond is for the cysteine-cysteine linkages, and hydrophobic interactions are for the clustering of the non-polar side chains which are away from water. Now let's see this question. Which of the following amino acids is not typically found in an alpha helix form due to its unique cyclic structure? Proline, glycine, arginine, or methionine. So it is saying that this kind of amino acids cannot form alpha helix structure because it is in the cyclic structure. So Fozia is saying A, Shivaswamy C, Aman is saying proline. Okay. So we have on A more, which is true also. So proline is basically having this structure like this. So in this is basically not typically found in alpha helix structure because of its unique cyclic structure. So like because of this, where the side chain is bonded to nitrogen atom. This one is bonded to nitrogen atom of the amino group and form a rigid ring. This ring restricts the rotation around the NC alpha bond and introduce a band in the polypeptide chain which disrupts the regular helical structure. Additionally, proline lacks the amide hydrogen, so necessary for forming the hydrogen bonding also it cannot form, that stabilizes the alpha helix structure. So in alpha helix we need this amide bound hydrogen group which helps in the formation of hydrogen bonding. So here we don't have any hydrogen uh, group here. No hydrogen bound group is no amide bound hydrogen is there. Also, it is not like its cyclic structure is very rigid and cannot be converted into regular helical structure. Like this is a helical structure, alpha helix, secondary structure of the protein. It forms a rigid right-handed spiral, like this right-handed spiral will be there, like this. While right-handed alpha helix is commonly found and each turn includes 3.6 amino acids, which is having the 5.4 angstrom bond length, and the form is stabilized by a hydrogen bonding. This is the hydrogen bonding which stabilizes this alpha helix form. So amino acids that disrupt an alpha helix basically includes the proline and glycine. So proline and glycines are, proline though is like it is not going to form uh, because of two reasons. One is because it cannot form the hydrogen bonding also, and another is it is as having the rigid cyclic structure, so that will not going to form that alpha helix structure. Also, the glycine is less commonly it will form alpha helix because of its flexibility issue, and it does not inherently disrupt the helix like proline does, but it is little less flexible. Because of that, alpha helix structure cannot be formed by glycine also, whereas arginine and methionine, they are structurally they are profound or compatible with the alpha helix structure and do not cause disruption, so they can exist in the secondary structures like alpha helix. So this is the glycine molecule. So it is having this amide H also, and this open ring structure also, same in methionine here also, and arginine if we talk about here, and this is amide, right? So this one is like, so they can form here because, but proline cannot because of the ring structure. So next is, which of the statement accurately defines Zwitter ion? What is Zwitter ion? An ion carrying an overall negative charge. A molecule possesses both positive and negative charge, including or resulting in no net electrical charge. Third is a molecule that only forms at extremely high pH level, or a molecule that is positively charged at low pH values. Anyone who would like to answer which is Zwitter ion? So most of us are saying B. So you guys are correct. Zwitterion basically is a molecule that is having both the positive charge also and negative charge also on different functional groups within the same molecule, but overall the net electrical charge will be 0. So this is the characteristics of the amino acid at their isoelectric point when where the amino group is protonated and the carboxylic group is deprotonated. A zwitterion does not have an overall negative charge. So the zwitterion can form at moderate pH levels but not at extremely high pH levels. At low pH level, the molecule will be at the positive charge due to protonation of both the groups, eliminating the ballast required for a zwitterion. So like this is a zwitterion. One group is positive functional group and another is negative functional group in the same molecule. And it varies based on the pH level. So if the pH is high, so anionic negative charge will be there. And if pH is very low, so positive or cationic property it will show. The isoelectric point of the protein is the pH at which the protein has a net electrical charge, positive, negative, no, or variable. Anyone would like to answer what is isoelectric point of a protein? C. Shivaswamy, no charge. Aman also C. No charge. Okay, let's see. You guys are correct. No charge. So in isoelectric point of the protein at a specific pH. So these are the terms like zwitterion or the isoelectric point. They all are relevant with the pH term. So specific pH should be included. So at a specific pH, so proteins has no net electric charge, meaning the total number of positive charge is equal to the total number of the negative charges. So at this particular pH, the protein does not migrate in an electric field as the charges cancel out each other, rendering the protein electrically neutral. So if pH is below the isoelectric point, so less than isoelectric point, so what will happen? The protein gains more protons and becomes a positively charged. And if pH is higher than Pi, so what will going to happen? The protein loses the protons and becoming the negatively charged. Thus, the concept of Pi is crucial for understanding the protein solubility, stability, and behavior in different environments, especially in processes like electrophoresis and protein purifications. So like this, when Zwitter, so we have this kind of uh, like when at different pH values or pK values, the behavior is changed uh, here. So we can say, so I am taking one example of alanine. So this is a titration curve of alanine. So at pH this, in this range, it is at its isoelectric point, like around maybe 6, and when it is at low pH, it is showing some kind of different behavior, and at very high pH, it is showing some kind of different behavior. So at low pH, it is showing the overall positive effect, and greater than 10, it is showing overall negative effect, but at pH 6, it is electrically neutral. So this region is basically known as isoelectric point. This is at some pK1 and this is some pK2. So this degree of ionization of a group at any given pH of the solution basically determined by this equation. Henderson Hasselbalch equation. So this basically equals to pH is equals to pKa plus log conjugated base upon concentration of conjugated acid. So basically, the Zwitter ion concept and isoelectric one point of the proteins, they basically both dependent on the pH. So at some pH they'll be neutral, and at high pH or low pH they'll have either the positive or the negative charges. So next question is, which of the following statements about protein denaturation is correct? Statement 1 is denaturation always involves breaking covalent bonds. Second is denatured protein can have increased or decreased solubility. B is correct. Okay, only two. So you guys are correct. So protein denaturation basically refers to the process where basically protein loses its native structure to the disruption of non-covalent interactions such as hydrogen bond, ionic bonds, and the hydrophobic interactions. And it does not typically involve breaking of the covalent bonds like peptide bond, which remains intact. So this is the wrong answer, because it only in the denaturation only non-covalent bonds will going to be break, and during denaturation protein may become either more or less soluble. Basically it will depend on the nature of the denaturing agent and the protein's environment. So it can even increase the solubility or decrease. So only two statement is correct. For example, if I am having the egg white protein and I am heat denaturating it, it will form aggregate and becomes insoluble, while some proteins will become more soluble when unfolded in certain solvents. So the denaturation basically is usually, this process is reversible, and the proteins can regain again their structure, which is known as renaturation process. Like this is the native structure, and it can go under folding, and this will become the inactive form it can go, which is protein denaturation. So denaturing agent can be physical and chemical. Physical means if I'm giving heat, or I'm stirring, or I'm shaking, I'm providing high pressure or ultraviolet radiation, that is all physical agents or denaturing agents. And chemicals are like pH, adding 6, 8 urea, or other compounds, SDS, synthetic detergents. So initially, if we so basically if we see the energy representation of the protein molecule with its conformation, so in denatured state, if we see like this, so native folded state, its energy, it's more stable basically. So let's see the melting point of, now let's discuss about fatty acids. So we are discussing the melting point of fatty acid as a uh, so it increases, decreases, remains unchanged, or becomes unpredictable when the number of double bonds increases. I hope so far all the questions are clear to you guys. Is there any doubt so far? Please let me know. So Aman, what do you think? Yes, ma'am, absolutely. Okay, so what do you think? What is okay? Today's session is very good. Okay, thank you so much for your feedback. If you want to share some things which I need to improve, also you, all of you, not just Aman, all of you can share your point of view. Also, please try to give this answer. If we are increasing the number of double bonds in the fatty acids, what will be the effect on the fatty acids? Points discussed elaborately, ma'am. Okay, thank you so much. Shiv Swami, thank you for your feedback. Okay, Anjali is saying this question answer is B, decreases. Also Shivaswamy and Aman B. So okay, we are locking B answer then. Let's see what is the correct answer. So B, you guys are correct. So what is happening when we are increasing the number of double bonds? So the melting point of fatty acids, it decreases due to the structural impact of these double bonds. So in the saturated one and the unsaturated one, we can see in the when we increasing the number of the double bonds, fatty acids melting point, it will decrease. So the double bond in fatty acids create kinks or bands in the hydrocarbon chain, which prevents the molecules from packing closely together in a solid form. So this loss in the packing efficiency or packing density reduces the intermolecular forces, primarily van der Waals interactions, making it easier for the fatty acid molecules to transition from a solid to a liquid state. So as a result, if more double bonds are there in a fatty acids, so what will going to happen? We will have lower melting point. In contrast, if we see in the saturated fatty acids, basically will have no double bonds and mostly they are the straight chains or packed tightly. So their melting point will be higher. So if we increase the double bond, which is the level of unsaturation, if we are increasing, so they will not be packed efficiently, or the the density of the packing efficiency will be lesser. Because of that, the melting point will be decreased. So basically fatty acids are classified into two types, like the saturated fatty acids and the unsaturated fatty acids. And the saturated fatty acids basically includes stearic acids, palmitic acids, mesteric acids, and the lauric acid. So stearic acid is like like beef, lard, and tallow is the example of stearic acid. Then palm oil, butter, and lard having the palmitic acid. Then coconut or the butter, they are having mesteric acid, and palm kernel, coconut, they have the lauric acid. Then in unsaturated, we have the MUFAs and the PUFAs, which are monounsaturated fatty acid and polyunsaturated fatty acid. So the MUFAs, they have the oleic acid, which is having which is present in the olive and the pecan oils, and which is also known as so it is sometimes we say omega 9 also, but not usually. We say oleic acid only, monounsaturated one. And PUFA has omega 3 and omega 6. So omega 3 is basically alpha linolenic acid, and then EPA and DHA. So basically ALA is basically found in the soybean and canola oils, and EPA is basically in the salmon, cod, cod liver oil, and DHA is basically in the salmon or the tuna fish, like that. And omega 6 is having linoleic acid, GLA, which is gamma linolenic acid, and AA is a arachidonic acid. So basically, these oil, linolenic acid is basically in the meat or safflower. GLA is basically in the vegetable oil, and AA is basically in the poultry and eggs. So now I am going to discuss about these fatty acids. So the omega-3 fatty acids, alpha-linolenic acid is converted into converted into which form basically in human body. So will it convert into ALA or DHA or oleic acid or palmitic acid? Shivaswamy C, oleic acid. Aman A, ALA. Okay, let's see. So here P answer is the correct, which is DHA. So alpha linolenic acid, which is ALA, which is basically this one, which is in one of the omega-3 acid, this one, which we get from the soybean and canola oils. So this is basically an essential omega-3 fatty acids that cannot be synthesized by human body. That's why we are known as this is the essential fatty acid, because our body cannot synthesize it. But it is very important for our diet. Once ingested, then this ALA will going to metabolize into series of enzymatic reactions to produce longer chain omega-3 fatty acids such as EPA and DHA. So this DHA is also very crucial for the brain functioning, eye health, and anti-inflammatory process. This so if we see here this alpha linolenic acid, so is basically it is converted into EPA, DPA, major compounds. I am just talking about the major compounds, and DHA. So basically, it when we consume this alpha linolenic acid, it will go under some kind of metabolic reactions from and with different, we can see here different enzymes are there. These are the different enzymes which will convert it into DHA as a final compound. So this DHA is very important for our brain function, eye health, and anti-inflammatory process. So that's why in some kind of oils which we sell in the market, it is say that DHA fortified, omega-3 fortified, some like fortificate. These oils will be fortified with these all these kind of compounds. So they market these oils like that these days, so that consumer can understand that okay, this is the essential fatty acids our body need it, so we can buy this kind of products. So DHA is the correct answer here. Let's discuss this next question. So we have in column 1 some fatty acids like alpha linolenic acid, then linoleic acid, then this arachidonic acid and eco eicosapentaenoic acid. So they have this functions in column 2, like precursors for omega-6, precursor for omega-3, and important for brain and heart health, and essential for cell membrane structures. Anyone who would like to answer this question? Okay, Shivaswamy and Anjali, D answer. Okay. So B answer is correct in this case. Let's see. So alpha linoleic acid, ALA, is basically crucial for brain and heart health because it is a precursor to the omega-3 fatty acid, EPA and DHA. So we discussed that ALA is basically the precursor for EPA and DHA, or that is it's very important for brain and heart. So it supports basically the cardiovascular and the neurological functions. So here we discussed this whole pathway here. And next is the linoleic acid. So it is basically essential for the cell membrane structure as it helps maintain membrane fluidity and integrity, particularly in phospholipid bilayers. So this is for the cell membrane structures. And this EPA is basically the precursor of omega-3 eicosanoids. So basically, which is basically anti-inflammatory roles it plays, which have anti-inflammatory effects and play roles in reducing the cardiovascular risks. And this AA is basically the precursor for omega-6 eicosanoids. So basically, it is good for the function in the inflammatory and immune responses. So if we see here, alpha linoleic acid, it is for the important for brain and heart, and linoleic acid is for the essential for cell membrane structure. Then this EPA is for the omega-3, and this AA is for omega-6. So if we see here, so this is the linoleic, which is 18 carbon and 2 double bonds, and linolenic acid 18,3. So this is also 18 carbon chain and 3 are the double bonds. And this AA is having 20 carbon chains and 4 double bonds, and EPA is 20 carbon chains and 5 double bonds. So we can see here different fatty acids, and it is basically the fatty acids are basically dependent on their structure, their function. So usually we in uh, human body we need 18 to 20, 18 and 20 carbon chain fatty acids. So 18 with one double bond, two double bond, and three double bond. So 20 with four, five, and six double bonds. So here we can see ALA is being converted into DHA. It is also the precursor for EPA, whereas omega-6, which is linoleic acid, it can be, we can obtain AA from LA. So this AA, DHA, EPA, these can be classified as here. If we see here, we classified in this table, in this basically here, these are the conditional essential fatty acids. So these are the conditional essential fatty acids. So they are essential, but they are produced in the, can be produced in the body but with the help of essential fatty acids. So our body must eat from the plants or from the different oil sources, fatty oil sources of from the outside, from the human body. So we need ALA and LA because they are the essential fatty acids, while others can be synthesized in the body. So they are conditionally essential. They are essential, but they can be synthesized in our body. So topics we discussed today are basically related to water and ice and its structure and the property. And we discussed about what happens if we add the solute, so that freezing point decreases. Then we discussed about free and the bound water and the water activity and sorption isotherm and molecular mobility and food stability. Then we discuss about the carbohydrate or the starch and the oligosaccharides, where we discuss about basically we discussed about the polysaccharides, and we discussed about the monosaccharides like glucose or fructose, and we also discussed about cellulose. So we discussed about starch, cellulose, and pectin in detail. So, and we also discussed about the retrogradation of starch, and we discussed about pectin and its gel forming properties. And we discussed about proteins and the polypeptides. So we discussed about different amino acids, and we discussed about which amino acids can form alpha helix or secondary structures, and which amino acids cannot form secondary structures. Then we discussed about the different bonds like hydrogen bonding or hydrophobic interactions or ionic interactions or sulfide bonds between different amino acids, and we discuss about the denaturation of the protein also. Then we discussed about the fatty acids and different fatty acids, like we discussed about basically we discussed about the essential fatty acids and the conditionally essential fatty acids. So this how they metabolize, and like that we discuss. And in the next week we'll be discussing about vitamins and minerals and phytochemicals and bioactives, then pigments and colors and the flavoring compounds. So you'll get all the material on the respective platforms, and if you have any question, you can post or you can ask now. So thank you so much for joining today's session and providing your valuable feedback to me. Please join the next session on the same time, 4 to 6 on next Sunday. So let's meet upcoming Sunday on the upcoming Sunday. So this is the end of the session for today. So thank you so much for joining the class. And if you have some doubt, you can stay, otherwise you can leave the meeting. So I'm turning off the recording.